Okay, guys, so this week we're going to talk about blood and then the heart. They're both pretty closely connected. Next week we'll talk about blood vessels and the lymphatic and immune system, also all pretty interconnected. Remember, we learned that blood is a connective tissue. What makes it a connective tissue is its fluid matrix. The fluid matrix of blood is the plasma. And when blood is put into a test tube and into a centrifuge and spun down, then you can really see the separation of the elements. Plasma is the clear, the extracellular matrix. It accounts for probably about 55% of your blood. Most of plasma is water, but it also contains some proteins like albumin, some nutrients, some electrolytes, some hormones, and some gases. It plays a role in blood clotting, plays a role in the immune system, and in the regulation of fluid volume. Plasma without the clotting proteins is called serum. And then the formed elements, and you can see this on this here, this part on the bottom. The formed elements include the red blood cells, also known as erythrocytes, the white blood cells, known as leukocytes. They make up, and the platelets. They make up about the other 45% of the blood. Red blood cells are the heaviest of the formed elements. They sink to the bottom of the sample. You've all heard of blood tests called hematocrit. That is really a measure of the percentage of red blood cells in a sample of blood. The white blood cells and the platelets then form the narrow buff-colored band just underneath the plasma. This right here, you can see that's called the buffy coat. They only constitute maybe about 1% of the blood volume. And the combination of the plasma and the blood cells determines the viscosity or the stickiness of blood. The formation of blood cells is called hemopoiesis. All blood cells are made, all different types are made in the red bone marrow. You're going to find the red bone marrow in the ends of the long bones and in the flat, irregular bones like the sternum, the cranial bones, the vertebrae, and the pelvis. In the long bones, you find it mostly in the femur. Lymphatic tissue is what produces the lymphocytes, one of the white blood cells. And the lymphatic tissue is found in the spleen, the lymph nodes, and the thymus gland. The job of red blood cells is to deliver oxygen to the cells and take back carbon dioxide. A red blood cell is a disc, kind of like a disc shape. It's got a large surface area. Part of it is concave, which increases the surface area. Red blood cells lose almost all of their organelles during development. They do not have a nucleus or DNA, so they can't replicate themselves. That's why they're made in the red bone marrow. And the cytoskeleton of the red blood cell is stretchable fibers. It makes it flexible. That allows it to fold and stretch so it can squeeze through the tiny capillaries and then go back to its shape. More than a third of the interior of a red blood cell is filled with hemoglobin, and the red pigment of hemoglobin is what gives blood its color. Hemoglobin consists of 4 ribbon-like protein chains called globins. You can see it's pointing them out to you here. They actually pictured them very nicely in different colors for you. And then bound to each globin is a molecule with iron in it called heme. That's where we come up with hemoglobin. Each heme molecule, each one of these little molecules right here, can combine with one molecule of oxygen. So one hemoglobin molecule can combine with 4 molecules of oxygen, and that forms oxyhemoglobin. And if you break the name down, using your medical terminology, it means hemoglobin with oxygen. Hemoglobin also carries carbon dioxide, but the carbon dioxide binds with the globin, not the heme. How much oxygen the blood can carry depends obviously on the number of red blood cells and the amount of hemoglobin it contains. Red blood cells live for about 120 days. I always think of that as it's pretty close to the same as a college semester. So at the beginning of the semester, you start out nice and fresh, and by the end of the semester, you're all broken down, worn down, stressed out. Same thing happens with your red blood cell. After about 120 days, red blood cells will then die, they'll break up, and they get consumed by phagocytic cells in the spleen and the liver. And I know we learned about the phagocytic cells in the liver a few weeks ago. your body is constantly producing red blood cells to maintain homeostasis. And this is a process called erythropoiesis, E-R-Y-T-H, remember, refers to red. It's maintained through a negative feedback loop, like a lot of things in your body. If the oxygen levels decline, because that'll go down as the damaged red blood cells are removed from circulation. The kidneys will detect this. They'll secrete erythropoietin, a hormone. That erythropoietin will stimulate the red bone marrow to make new blood cells, new red blood cells. Immature erythrocytes, and these are called reticulocytes, are released into circulation, and after a couple of days, that reticulocyte has matured into a red blood cell. That increased number of red blood cells causes oxygen levels to increase, so less erythropoietin is secreted by the kidneys, and then the red blood cell production declines, and this is a constant back and forth. Just like when we learned about the acid-base balance is a constant back and forth, so is this. Macrophages in the liver and the spleen will ingest and destroy the old red blood cells, and in the process, the hemoglobin is broken into its two components, the globin and the heme. Globin is broken down into amino acids, and then they're used for energy or to create new proteins, and the heme is broken down into iron and bilirubin. Iron is used to create new hemoglobin, and bilirubin is excreted into the intestines as part of the bile. When your body breaks down too many red blood cells, then the body can't assimilate it can't take care of the extra bilirubin that's floating around. This extra bilirubin enters the tissue, and that's what causes the skin and the sclera to take on a yellowish color called jaundice. This also happens if your liver fails and doesn't break down the red blood cells or breaks them down too much. White blood cells are called leukocytes. These are the fewest of the formed elements. And your body contains 5 different types of white blood cells. They're all different in size, appearance, number or abundance, and their function. All of the leukocytes contain a nucleus, and they contain other internal structures that look like granules when stained and examined under a microscope. Granulocytes have obvious granules, and a lot of them. Agranulocytes have very few of them, or maybe even sometimes none at all. Granulocytes have a single multilobar nucleus, and there's 3 types of granulocytes: neutrophils, eosinophils, and basophils. And then the agranulocytes are the lymphocytes and the monocytes. There is a chart in your text that details all of this. I'm not going to expect you to know exactly what each type of white blood cell does, but I do want you to know the different types and how they are different from each other. Platelets. Platelets are also called thrombocytes. They're the second most abundant of the formed elements. So red blood cells are the most, platelets are second, and then the leukocytes are third. Platelets play a key role in stopping bleeding, and stopping bleeding, the process of that is called hemostasis. They're actually fragments of larger bone marrow cells. The edges of these bone marrow cells, called megakaryocytes, break off and form the cell fragments called platelets. And platelets only live about 7 days. Hemostasis, what happens when you injure yourself, you get a cut, a blood vessel is cut, your body's got to react pretty quickly to stop the flow of blood. It does so. There's a whole sequence of events that happens, starting with a vascular spasm, and then it forms a platelet plug, and then the formation of a blood clot. So as soon as a blood vessel is injured, the smooth muscle fibers in the wall of the vessel go into a spasm. That slows the flow of blood. So you can see that's got it pictured here, this injury, and the walls are going into a spasm to stop the blood from flowing out. The break in the blood vessel exposes collagen fibers, creates a rough spot on the vessel's normally very smooth, very slick interior, and then this rough spot triggers the passing platelets to go from their normal state into sticky platelets. The sticky platelets then stick to the vessel wall and to each other and form a platelet plug. The platelets facilitate clumping by secreting several chemicals. Some of these chemicals that they secrete causes the vessel to constrict even further, and then others of them attract even more platelets. This platelet plug forms a temporary seal in the vessel wall until the clot forms, which is the more stable solution to that. And of course, forming a clot, there's not just one way to do it. There's a couple of different ways. There's two different pathways. We call it the extrinsic pathway. That is started by damage to areas outside of the blood, like from outside the body. And then the intrinsic pathway is started by factors inside the blood. Both of them end with the formation of factor X. Once factor X is formed, then it starts the common pathway, which begins with the production of an enzyme that's called the prothrombin activator. The prothrombin activator then works on a globulin called prothrombin, kind of makes sense, doesn't it? Which converts that into the enzyme thrombin. Thrombin transforms the soluble plasma protein fibrinogen into fine threads of insoluble fibrin. The sticky fibrin threads form a web at the site of the injury. Red blood cells and platelets flowing through the web get caught. They create a clot of fibrin, blood cells, and platelets, and a blood clot can then effectively seal breaks in a smaller vessel. A larger vessel, obviously, may not be enough to stop the bleeding. So it has been formed, and then we need to dissolve that blood clot. Soon after the blood clot forms, platelets trapped within the fibrin web contract. They pull the edges of the damaged vessel closer together. After the vessel is healed, then there's a chain of reactions that converts plasminogen, which is an inactive plasma protein, into plasmin. Plasmin then dissolves the fibrin meshwork and the clot breaks up. This is what we call fibrinolysis. OLYSIS is a suffix in medical terminology that means the breaking apart of or the dissolution of, and fibrin obviously refers to fibrin. Some things that can prevent blood clots from happening. The first one, the smooth endothelium. The inside of your blood vessels has to be smooth. That layer of epithelial tissue that's inside your blood vessels needs to be smooth so that the platelets don't think it's a break and start to stick to it and make a blood clot. Because we don't want unwanted blood clots inside your blood vessels. We only want clots if there's been a break in them. Normal circulation or blood flow, as long as the blood keeps flowing, it's not pooling like when it pools in your legs, like when you're sitting in class for hours on end. When it pools like that, then the thrombin can accumulate and lead to a clot formation. That's why you hear people who've been on long trips, sometimes they wind up getting admitted with a deep vein thrombosis or a blood clot in the vessels of their legs. And then there's some medications, some anticoagulants that can help prevent all of this process from starting. So let's talk about blood types for a few minutes, the different blood types in the body. Most of you should be familiar that there are really 4 different types. There's type A, type B, type AB, and type O. These are determined by antigens that are on the surface of your red blood cells. And the surface of each red blood cells, the antigens are called a protein. There's really 2 types of antigens, type A and type B. You can see on this picture here, they've got both different types pictured. I don't know if this is really what they look like or this is just the shape they decided to use to help to tell them apart. So if you're a person with type A blood, then you have this type A antigen on the surface of your red blood cells. Same as type B, you have this type B antigen on the surface of your red blood cells. If you're a type AB, then you have both of them. And if you're a type O, you don't have either of them. So your blood cells carry the antigens, and your blood plasma carries antibodies against antigens of other blood types. So type A blood has B antibodies, and they pictured them here in different colors. I don't know if that's more confusing or less confusing. So type A blood is going to have anti-B antibodies. Type B blood has anti-A antibodies. And type AB blood doesn't have any antibodies, and type O has both types of antibodies. When you receive a transfusion, if you're someone with type A blood and for some reason you wind up getting type B blood, your type B antibodies that you have right here are going to recognize the type A blood that you have, but they're also going to recognize the type B blood that you're giving as something that shouldn't be there, and they're going to attack. They're going to attack and cause that type B blood to clump together. It's called agglutination, and that could cause all sorts of problems inside your body. You're going to have a transfusion reaction, which can be really serious. These blood cells can clump up black blood vessels, cut off the flow of oxygen. The red blood cells can also burst and release their hemoglobin into the bloodstream. This free hemoglobin can block tubules in your kidneys, which can lead to failure of your kidneys. It could actually be a fatal reaction that you could have. And we're going to do an experiment in the lab where you're going to test some synthetic blood and try to figure out what blood type it is based on the reaction you see. We're going to talk about for blood is the Rh group. You've probably heard people say, oh, I'm O positive or O negative, A positive, A negative. You also, besides the A, B, and O types and the AB type, you also have positive and negative for each of them. Rh positive blood contains the Rh antigen, and the Rh negative blood does not have that antigen. So if you were somebody who is an Rh negative and you get Rh positive blood, you're going to have another reaction, same as if you're an A type who gets B type blood. Because your body sees that Rh positive as a foreign body and something that needs to be attacked. Normally you don't contain the anti-Rh antibodies, and you don't get them until you get your first transfusion of that. So if you're A negative and you receive A positive blood, you're not going to have a reaction with that first transfusion. But if you were to get another transfusion of A positive blood, you're going to have a reaction because your body has developed the antibodies in relation to the first transfusion you got. And then as far as pregnancy goes, the same thing can happen pretty similar if a woman who is Rh negative conceives a baby who is Rh positive because the father is Rh positive. Normally mother and baby blood does not mix. So the first pregnancy with an Rh positive fetus is going to proceed normally. During delivery, sometimes the fetus's blood mixes with that of the mother at just a very tiny amount, and that introduces those Rh antigens into the mother's bloodstream. So then the mother has formed the antibodies against that foreign substance. So if the mom gets pregnant again with another Rh positive baby, even though the blood is not mixing, the mother's body is going to recognize that as foreign, and it's going to attack. These anti-Rh antibodies can pass through the placenta and attack the fetal red blood cells, causing agglutination, breakdown of the red blood cells, and then the infant develops a severe anemia called erythroblastosis fetalis. That could be fatal to the baby. It's definitely very serious, something we don't want to see happen. So we've talked about the different components of blood, the red blood cells, the hemoglobin that travels on the red blood cells carrying oxygen and carbon dioxide, the white blood cells, the 5 different types of them, and the platelets. We've also talked about hemostasis, the formation of the blood clot, and then we talked about the blood types and the antigens and antibodies involved in that. A lot of information in a short period of time. Hopefully our lab will help to explain some of that more to you.